favorite is when it takes forever to come up in my mind. You guys are probably watching it, so I'm going. Okay, guys, let's learn something. So we're almost there. We're almost at the end.
Yeah, 17 more days.
How many? 17?
Including weekends?
Oh, including weekend.
It's like 13 more.
No, I gotta have 17 lectures for you. I believe in my mind it's about, what, 1, 2,
We have
3.
4 lectures. 3 lectures.
3 lectures left. 2 exams. So this next exam is going to be like the equivalent of your final because it's fairly comprehensive and cumulative if you think about it. You can freak out if you want, but like, you all knew that this whole course built on itself. So, and then the final is going to be like on 2 lectures. So it's weird how that works.
Get hundreds from there.
Yeah, so I'm going to be giving out hundreds to everyone.
You don't even need to write it.
I'm not, yeah,
Just go straight.
I'm just gonna go straight down the road.
Exactly.
Exactly. Okay, some quick business. Exam 3 is next week, which leads to this week's recitation is review. Some of you guys have seen that already. No Dr. Texel, just me, come to my recitations. I've already posted a worksheet, some like stuff on Blackboard. Feel free to try to work on that first. Today's recitation had the unfortunate experience of not having that too far in advance. And so it might work best if you get through the whole list of stuff before you even come on Thursday. And then of course, for those of you who came today, feel free to come again on Thursday if you really love it so much. And then there's a few like big picture type of practice problems. Next week, it was overwhelming vote that we should do recitation online with the option of coming to recitation for help if you need it. And so that's what we're going to do. So you don't have to come, but I will be there at my normal times. If I'm sitting in an empty room, then I'll probably go to my office. So if you're like, oh my God, I was going to come, you weren't there, just check my office. But I am available during our normal recitation times. If you need help getting the paper, if you're like, no, I need to study, I'm not going to get to this till Friday, that's fine. It will be available all week for you guys to do. Just don't forget about it, because that happened last time to a lot of people. Dr. Texel's class, again, does not exist next week either. So you guys didn't get to say your proper goodbyes to her, so you should stop by and give her hugs. Because you won't be seeing her anymore, apparently. Okay, so far this is what we've been doing. It's a lot of stuff. This is a pretty in-depth exam, apparently. We finished with talking just about the left operon and some examples of gene expression regulation. We talked about insulators, repressors, activators, how they're regulated. And so we're kind of going to just continue with this stuff today. Hopefully get close to the end of that, and that will leave Thursday for translation. And then I will do that. Maybe. Here we go. Okay, so we're talking about gene regulatory proteins. So that's activators and repressors. And so the role of activators is to increase the rate of transcription initiation. So basically what they're doing is they're helping to attract and position and modify the general transcription factors in RNA polymerase II at the promoter so transcription can begin. So they're acting on the transcriptional machinery, and they help with modifying chromatin structure around the promoter. That's a whole lot of words for saying they help to recreate general transcription factors and make it so that chromatin structure is conducive to transcription, right? What do I mean by conducive to transcription? That means more open, promoters exposed, not tightly wound around nucleosomes, And so this is where you're going to feel like, wait a minute, I learned this before, but this feels like a long time ago. And you're going to have to kind of like go back in time a little to the first few weeks of the semester, because what we're doing now is starting to tie a lot of it together. Okay, so don't forget about that stuff. Any questions about this? Major concept. So you're like, what is she talking about? Hopefully no one's thinking about it. So what I'm saying in picture form is that activators are activating transcription by recruiting RNA polymerase II whole enzyme complex, right? So that's, you're going to have your activator binding to this upstream DNA element, and it's helping to recruit all these other things, all your general, let's see, transcription factors and RNA polymerase and mediator, that's all getting recruited to your promoter. There's your TATA box. And when that all happens, you can have transcription. That's what your activator, that's a major thing that the activators are doing. Yes?
In that picture, it looks like everything's assembled and then attaches itself to the promoter. That's what happens.
Yes and no. So you'll have, obviously, your TF2D is going to come first. And so there is a bit of sequential stuff going on. I think really this is more just for the completeness of the picture, but there are some things that do assemble together at the same time. But the way to think about it is really recognition of your TATA box by TATA binding protein, because that's your big recognition of promoter signal. That has to happen first, and then you're going to kind of get assembly of all these general transcription factors and RNA polymerase, and then mediator, kind of all in a major second step, but there's still kind of sequential assembly going on. Okay? Okay, so that's one thing that might happen. An activator might also help just with the general assembly of general transcription factors. So here I said it's helping to recruit everything. That's one thing. But then also it might help with recruiting those general transcription factors. Okay, so here you're seeing your activator binding, and you're seeing it's helping to bring in TF2D, B, and A. And that's not, like, this is just an example. I'm not saying it always brings these three factors in. It's just assembly of general transcription factors. So another thing they can do is they can bind and recruit chromatin remodeling complexes, and they help to alter your chromatin structure, right? Remember we talked all about chromatin structure and function, and acetylation, and methylation, and HATs, and chromatin remodeling, nucleosome remodeling complexes, right? This is, remember, tying it together. People are always like, what's starting this all? And I was like, oh, transcription factors. Well, these are these transcription factors that we were talking about that kind of start the whole thing. So you'll have an activator that'll bind to the DNA. And so here you've got your promoter kind of all wound up in your chromatin. So you can't do transcription that way. You need to somehow open things up. And so then you'll get recruitment of your chromatin modeling complex, which will activate these histone chaperones to remove your nucleosomes and allow for exposure of your promoter. And then you can start to get gene expression. Yeah.
Did you just say gene activator protein is like a transcription factor?
Yeah.
Okay.
Yeah. So a transcription factor is a protein. Thank you for saying this, because I tend to use different words sometimes to talk about the same thing. So a transcription factor is a protein that's going to affect transcription, whether or not it will happen. And a gene activator protein or a gene regulatory protein, these are all proteins that are going to somehow interact with the DNA and affect whether or not transcription is going to happen. So you can use them interchangeably. Okay? Yes?
What is really happening in that picture? Is it like...
So what you're seeing here, let's walk through it, okay? So what you're seeing here is your gene regulatory protein binding, and then you see you've got your promoter obscured, so it can't be interacted with, right? And so that gene activator protein is allowing for recruitment of your chromatin remodeling complex. So here you see your nucleosome remodeling complex comes in and helps to move your nucleosomes. And that requires the activity of these histone chaperones that we talked about before. So remember we talked about sometimes you'll get your histones moved around on the DNA, sometimes they'll be disassembled and actually removed from the DNA. And so that's what you're seeing here. Your histone chaperones are coming in, removing your nucleosomes and replacing them elsewhere. But either way, notice now you have exposure of that promoter. And then you can also then get recruitment of histone modifying enzymes such as, what's your number one histone modifying enzyme for gene expression? HATS, right? So here, bless you. The squeaky sneeze. Okay, histone modifying enzyme, so this will be your HAT, and it's going to then bind to your activator protein and, for example, acetylate those histones. And then you're going to get those histone modifications that are consistent with activation of gene expression. And then you can get recruitment of your general transcription factors and mediator and RNA polymerase and activation of gene expression. Okay, does that answer your question? Any other questions? So hopefully most of you are going, oh, and it's just kind of tying two things together at this point, hopefully. So this again was in the last figure, but you can have activators recruiting HATs, right? Because there are some transferases that are then gonna, bless you, or whatever that's going on over there. You'll get acetylation, and then that can create. Remember, when you acetylate histone tails, you also make binding sites for recruitment of other proteins, and so that could then allow for recruitment of additional activator proteins. Because it's not just one activator. Usually there are many activators involved in activating gene expression. Okay? Yes?
So HATs can recruit mediators?
HATs can recruit, not directly.
Okay.
HATs can acetylate histones, right? And that can open up the DNA, and it can also recruit other proteins, such as coactivators. And maybe mediator. So think about that. Really what mediator is interacting with is your general transcription factors, right?
Yeah.
So a subunit of TF2D contains two bromodomains. So how might an activator promote assembly of general transcription factors at the promoter? Right, so I'm telling you, activators promote assembly of general transcription factors, activators do this, activators do that. Now I'm telling you that one of the subunits of TF2D has two bromodomains. So can you guys piece things together? Yeah.
Would it mean to stop the DNA from opening at the point where it binds?
So why don't you think about it for a minute. Before I repeat what you said, I'm going to give you a chance to take it back.
Take it back.
So bromodomain. So what does TF2D do?
It lands on the promoter.
Out of box, binds promoter. Does that activate or repress gene expression? Yes, it's usually involved in activation of gene expression, identifying the promoter, so your general transcription factors and RNA polymerase can all assemble there, right? Okay.
I mean,
Does it have something to do that bromodomain is associated with acetylation?
Right, so yeah, exactly. So bromodomains bind to acetylated glycines, right? We learned that. Right? Right. So I was telling you that activators promote HATs. And HATs do what? Acetylate. And now I'm telling you that TF2D has bromodomains. Right, so this is all, so remember I said activators help recruit general transcription factors. Activators recruit HATs. that acetylate histones, general transcription factors, have bromodomains. So you should start to see that actually what's happening here is you're getting acetylation of histone tails, which is helping with recruitment of general transcription factors. So I'll go through the whole thing, and you can tell me what you think, okay? So this is right out of the textbook, and this is a really nice figure that kind of helps to put it all together. It's very busy, but if you slow yourself down and you go step by step, it makes a lot of sense. Okay, so here we're connecting histone modification with transcription initiation. Right, so here's your histone with your DNA wrapped around it, and here's your promoter, okay? Now this promoter is available, it's not wrapped around histone. And so you'll have some transcription factor that's going to come and bind to some DNA elements upstream from the promoter. And maybe it will recruit a histone acetyltransferase, a HAT, right? And what a HATs do is they acetylate, right? So then that HAT's going to go ahead and acetylate histone tails on these different lysine residues, okay? And so then maybe that will create a binding site, because remember, acetylation of histone tails mostly creates binding sites for recruitment of other things. And so maybe the acetylation will help with recruitment, or you'll have the gene activator will help with recruitment of kinase, right? So then that kinase will then go and phosphorylate histone tails. We didn't talk too much about phosphorylation of histone tails, but we did talk about how it happens, right? And that again, when you get post-translational modifications, usually you're creating binding sites for things, right? So here you're starting to generate this histone code. Remember we spoke about this histone code as these different modifications that tend to be involved with some sort of outcome. And so you're starting to see the generation of a histone code that's involved with transcription initiation. And so then you'll get recruitment of a chromatin remodeling complex. And then you'll get recruitment of TF2D. Right? And remember I said TF2D has two bromodomains, so it can bind to acetylated lysines. So now you've got TF2D that's binding to histone tails that are modified in such a way that they'll recruit TF2D. And then you also have your tautobox, part of your promoter, which is going to bind to. And then that also allows for recruitment of the rest of the machinery for transcription, and then you get transcription. Yes?
So bromodomain binds to acetylated lysine on the histone tail?
Yep. Yep. So that's what we learned in the beginning of the semester, right? Bromodomains bind to acetylated lysines on histone tails. Yep. So now what we're doing is we're connecting that.
So that's the mechanism that works on the TF2D binding?
That's in this case, yeah. So that's one mechanism. So just to go through it once more with big picture, right? So you'll get an activator that's going to bind to some DNA element, and that's going to recruit a HAT that's going to modify your histone tails. And that's going to result in recruitment of a kinase, which is then going to further modify histone tails. Remember, what we're doing is we're modifying to create binding sites for the appropriate proteins. And so this is your histone code for transcription initiation. And then you're going to get further recruitment of HATs and then more modifications. So again, generating this histone code for transcription initiation. And once you have those tails modified in the way that's ideal for interaction with nucleosome remodeling complexes or TF2D or whatever, in this case, what we're seeing in this example is you're getting interaction with a chromatin remodeling complex and recruitment of TF2D. And those are two things that are going to encourage remodeling of chromatin structure for transcription and recruitment of general transcription factors and initiation of transcription. So this is really just coming back to what we started the whole semester with. And I was saying this So I was just teaching you little teeny bits at a time, but this is kind of everything coming together. Any questions about this? Does this make sense? A little bit? I don't know, I'm getting a lot of blank stares. I'm sorry. Huh?
It's a lot to take in.
Is that a lot to take in? Okay. You know it all, you just have to piece it together. It's like a puzzle where you've looked at all the pieces already, but now you just need to put them together. Questions? Yes?
What's the jobs of the Prometeo remodeling complexes, all my historians are like,
So what is the job of a chromatin remodeling complex? What does it do?
Moving the nucleus when it's open.
It moves nucleosome.
or why am I moving?
So is transcription going to work very well if your DNA is all wrapped around the histone? Yeah. Yeah, exactly. So you need to make it so that, so let's go back, just quickly look at this picture. Where was it? This one. You can see you get recruitment of these nucleosome remodeling complexes, and it kind of opens up that whole region there, right? And so then you'll get the modification, you'll get recruitment of your general transcription factors and RNA polymerase. Okay? Any other questions? Yes.
So do all the other transcription factors also have bromodomains, or is this one or just, yeah?
Do all the other transcription factors also have bromodomains, or is it just this one? I don't know. That's an easy one to look up, isn't it? I think because that this is the one that's kind of the first one that kind of identifies the promoter. My guess is you really just need that first one right there, and then the other ones will assemble. But it'd be interesting to look at the sequences, because you just pull up every single one on PubMed, not PubMed, but like GenBank or like Uniprot or any of those databases that look at protein sequence, and they usually pull out any domains they have, and you can see exactly what they have. I mean, you can also see from this figure that this also has a probably an SH2 domain or some kind of phosphorylated, what is it? Phosphorylated serine binding domain also. Good question, though. Okay, so what needs to happen for successful transcription? Everything we've been talking about, right? You need some kind of gene activator to bind, because, well, you need to know what genes are going to be transcribed, right? You're going to get recruitment of chromatin remodeling complexes. You need to open up the DNA. You're going to get histone modification, again, because you need to open up the DNA. Other activator proteins are going to get involved. Then you're going to get mediator in your general transcription factors binding, and then you're going to get RNA polymerase binding, and then you're going to get your pre-initiation complex at the promoter, and you can get transcription initiation. So that's kind of putting it all together. Questions? Okay. So I made this flow chart because, I don't know, it seemed like it's a good way to think about things. So when you're thinking about gene regulatory proteins and what they do, ask yourself, does this promote gene expression? Because if they do, they're most likely an activator, right? Activators do anything that promotes gene expression, from recruiting hats to recruiting general transcription factors. If no, if it doesn't promote gene expression, then ask yourself, well, is it actively inhibiting gene expression? And if yes, it's likely a repressor. And repressors do anything that you know to inhibit gene expression, such as recruiting HDACs, methyltransferases, or blocking assembly of RNA polymerase and general transcription factors. All the things that you already know are associated with repression. And that's usually how you can tell whether or not a protein is acting as an activator or a repressor. Okay? What do you think? Do you guys agree with me? Sure, if you say so. Okay. Okay, so let's talk about repressors. We've been talking about activators, and so this is exactly what you would expect. So I talked about activators interacting with HATs and chromatin remodeling complexes and all this stuff. And so you're going to see this similar kind of thing going on, right? So repressors can inhibit gene expression through competitive binding with activators, right? If they're binding in the same place and preventing an activator from binding, that's one way they can repress gene expression, right? They also can bind and interact with the activation domain of an activator. And then remember we talked about how activators are modular and they have a DNA binding domain, and they have a transcription activation domain, and the DNA binding domain binds to the DNA, and the transcription activation domain interacts with general transcription factors, mediator, RNA polymerase. Well, you can have a repressor that's going to bind right next to your activator and bind specifically to the transcription activation domain of your activator. So now it can't interact with RNA polymerase, general transcription factors, mediator, any of that. So that's also repressing gene expression, right? Then you can also actively prevent assembly of general transcription factors and recruitment of RNA polymerase, right? So you can have a binding site for a repressor that's going to interact with TF2D and prevent assembly of all the other general transcription factors and RNA polymerase. That's another way to repress. Notice in all of these, you're somehow preventing RNA polymerase from coming, right? That's the theme. It can seem like I'm giving you a laundry list, but it's just you can come at it from different angles. Yep.
What if repressors is topping up your activator? In terms of just preventing it from interacting, so it tops it into pieces. There's like one DNA domain, like the answer part of
So
it.
you're asking if a repressor can physically cleave
Yeah.
an activator. Well, one way for repressing gene expression could be to degrade your activator, right? And that's not going to be in this list, but every protein has a half-life, and if you shorten the half-life of your activator, then you'll essentially get repression, right? You can also get recruitment of chromatin remodeling complexes to do the opposite of what we were just talking about before. We're saying, oh, we need chromatin remodeling complexes to move your histones to open up the DNA to provide access to the promoter. But you can also recruit a chromatin remodeling complex to close up the DNA and remodel your nucleosomes so as that to decrease accessibility to the promoter. Yes?
This is all done through the
Repressors. All the different ways that repressors connect. There's a nice figure in the textbook that goes through this really nicely. So I highly recommend look at that figure. I mean, I took it right from there. And then read the associated text in addition.
So repressors have chromodomains.
Do repressors have chromodomains?
They bind.
Yeah, why not? So and remember we spoke about that when we learned about methylation and all that. We talked about how you have methylation and then it will recruit chromodomain containing proteins. And so some repressors can bind to methylated lysines on histones. Yeah. Any other questions? See, I can see that some people are starting to piece it together, which makes me happy. So also, repressors can do just what activators do, but the opposite, right? Recruit HDACs to deacetylate histones. So that's, again, it's repressing. And then also recruit histone methyltransferases. Again, methylating histones and repressing. So those are all different ways that activators work, all different ways that repressors work. And if you make a list, you can start to see that kind of like repressors act in the opposite way as activators. But they're both binding to DNA and recruiting other things, okay? And affecting the accessibility of the DNA for transcription factors binding. Questions? Okay. So you can think of eukaryotic genes as being regulated by a whole committee of proteins, right? It's not just one protein, not just one transcription factor or activator or repressor, but usually there's a whole bunch of proteins that are assembling. And I had mentioned this before. And it's not so much the protein itself that matters as the DNA sequence that's bound and where it's bound, and what other proteins it recruits. So don't worry too much about exact, like this protein is an activator, this protein is a repressor. Just think about what is the outcome on gene expression, and how is it affecting gene expression. So, and this is similar to what we've seen before. This is just showing that enhancer regions will bind a whole bunch of different proteins that assemble a large number of gene regulatory proteins to activate transcription. Not just one, but many. And you'll have this looping out and this architectural DNA binding protein that its whole job is to kind of bend the DNA and loop it out. Okay, so we just talked about repressors and activators and how that all works. That mostly deals with transcriptional regulation at the point of initiation, right? That's all talking about, will initiation happen or not? But you can have transcription regulated at many different points of this process, besides just initiation. It could be at elongation, and it can be at termination also. So I'm going to give you a few examples of those. And again, this is, I think this is all eukaryotic. Okay, so regulation of initiation, this is what I've been talking about till like on blooming phase. So there you go. Regulation of initiation. Does RNA polymerase get recruited to activate gene expression? Right? Remember, big picture, gene expression is often activated by some kind of signaling, which results in an activator entering the nucleus and binding to DNA. That's what's regulating your initiation. So this is just showing, in this case, you've got it's a growth factor receptor that's binding to some kind of growth factor, and then you have all this phosphorylation associated with signal transduction that results in activator proteins or transcription factors or gene regulatory proteins, however you want to call it, go into the nucleus, bind to DNA, activate gene expression. So these gene regulatory proteins themselves can be regulated, right? So this is still talking about regulation of initiation, but this is talking at the point about that transcription factor. I keep saying it gets in, it binds to DNA, and this is what happens. This is how it behaves. But don't forget that transcription factor itself is regulated. There needs to be some way to control whether or not it's ever going to get into the nucleus and bind to DNA, right? Otherwise, we'd have all gene expression all the time, right? So this is seven different ways that you can regulate gene expression. Regulate the activity of an activator or gene regulatory protein. I shouldn't even have to tell you these. Actually, I'm going to make it go away. And I want you guys to come up with different ways that gene regulatory proteins are regulated. Okay? Now, so before I start taking answers, I want to explain a little bit more what I mean to make sure the most people in this room can participate. Because I always have like 5 or 6 people who are like, I know. And part of it is because they're just like really excited to raise their hands. But the rest of you guys I know also, but maybe like are just a little slower to understand the question or what I'm asking. So we're going to take a minute here and just talk for a second. Okay? So I'm talking about you know, these different proteins here, these gene regulatory proteins, right, like this one here, it's going to go in and bind to some enhancer region, activate gene expression. But that needs to be regulated. If it always went into the nucleus and bound to DNA, well, what would be the point of regulating gene expression? It would just be like all gene expression all the time. So I want you guys to think about, well, which ways, what kinds of mechanisms can you come up with that might control whether or not this protein goes into the nucleus and activates gene expression. So if you want, we'll make this like a stop and chat with your friends moment. Okay. Now, some of you guys might have the lecture in front of you on PowerPoint and are like, oh, you know all the answers. Fine. That's fine. But I want you guys to stop and think for a second. So take 2 minutes to think in your head, how might a gene regulatory protein, its activity, be regulated? Okay?
So you're talking about that one specific, not like the other ones, right?
This one, or, you know, anything that's going to go, these three, let's say these three. I'm not saying look up Krebs, CFOS, and ELF, no. I'm talking about, like, generic gene regulatory.
Oh, okay.
Yeah. And it could be the way that you might think about how you might regulate any protein. What?
Or like transcription factors, activators, gene regulatory proteins. It's all the same thing.
Yeah, sorry.
No, I just.
So general.
No, so general
These are like a specific.
Yeah. And then there's your activators.
So there's something actually binding to it and something.
But you know, I haven't been sick all semester. That's a record.
Because I'm scared.
Because everyone gets sick. It's like the worst thing ever. It's worse than before. I really like the recipes for projects that sit whether or not it's some pollen pride. So they're only if they're pollen
Okay. I think that everyone has like end of semester, the end of semester blues. You guys are all sad that it's gonna end, you know? And you're like, it's kind of like you don't want to tell me that you're learning things because you want me to be there even longer for you. You're like, I don't know what I'm doing. Make the semester last longer.
No.
You're
I love them, but no.
we can look into this straight through the summer.
No.
You're all
There's somewhere outside of the
Okay, so I explained the question a few times to a few different people, which is basically what is regulating the regulators? What kinds of mechanisms can you think of that are going to control whether these proteins can get in there and bind to and activate gene expression? So who had some ideas? Yeah.
Well, a signal, like activators are gonna have to somehow get to the nucleus via ligand. So if the ligand, like if the cell is not showing that ligand, then the cell won't be able to get in. I mean the signal. And if like this ligand like has it in it, like it's blocked, and then the signal won't be able to get in. And start transcription.
Okay, so I'm hearing a bunch of things. So you're talking about this ligand up here?
No, I'm talking about like that.
So are you talking about the transcription factors?
Yeah. But it doesn't like mean, doesn't have like, it's not like automatically admissible into the.
Okay, so you're talking about, okay, there's some way that these proteins are regulated whether they can go into the nucleus.
Yeah.
Okay, so you're saying regulation at the point of nuclear import.
Yeah.
Okay. So who knows how proteins get in and out of the nucleus? We didn't learn it in this class, but we can talk about it for a second. Yep. Right, so she said NLS, nuclear localization signal. So normally what happens, there are nuclear pores in your nuclear envelope, and it's kind of like a gate that the proteins have to pass through, and they need to interact with nuclear import receptors. And so if you have a nuclear localization signal, then it will bind to a nuclear import receptor, and then it will bring it through the nuclear port into the nucleus. And so you're right. You actually, whether or not you can see your NLS will determine whether or not you can get into the nucleus. So yeah, not everyone in here took cell biology, and that's fine, but you're able to know that whether or not you're in the nucleus or not determines activity at one level. So that's one thing. And so a lot of signaling pathways keep the transcription factor in the cytoplasm until it's activated somehow, either through degradation of an inhibitor or through phosphorylation or some protein-protein interaction, and then it can go into the nucleus. Okay, so one is cellular localization. Great. Okay, that's one mechanism. Yes?
Can we say if post-transcriptional modifications don't happen, maybe
Post-transcriptional or post-translational?
post-transcription?
So modification of the RNA or of the protein?
No, of the RNA.
Okay.
Before they make the protein.
Ah.
Wait.
So you're saying that this protein,
Yeah, like
before it's even made into protein. So you're saying, so you're saying maybe the protein's not even being made.
Yeah.
Okay, so that is one, right? So if there's no transcription factor, can you activate transcription? No, right? So if this protein is never made, it will never be able to bind to and act as an activator. So now we have two, right? One was nuclear localization, the other is gene expression. Yeah.
What if there's some regulated translation of that?
And that's the same thing. So if you're regulating translation of the protein, so basically different ways to control whether the protein will ever be made, either at the transcriptional level, translation, gene expression, all of those. Yeah. So it's either there or it's not. It's either in the nucleus or in the cytoplasm. Those are two mechanisms. Yeah.
What about my brain's kind of fuzzy when it comes to this, but like GTP and GDP.
GTP and GDP.
Like some need to some proteins when they need it to be catalyzed in order
Okay. So you're thinking about nuclear import, probably, because GDP and GTP control whether or not protein's gonna get into the nucleus.
Would that work as a?
Yeah, I mean, nah. If you're thinking about nuclear import, yes. If you're thinking about something else,
Okay.
I have a question.
yes.
The availability of DNA and the structure of the binding site.
The availability of DNA and the structure of their binding sites. So here you're going in a different direction. Definitely availability of DNA and structure of binding sites. All those, so availability of DNA, which DNA? The DNA of the gene that's going to be expressed, or the
Yes.
DNA encoding this protein?
No, the gene that's going to be expressed.
Right, so definitely that's what we've been talking about the whole time, is whether or not that DNA is available. That's regulation of initiation. But here we're talking about regulation of the transcription factors activity.
Oh, I flipped it. I'm sorry. That's what I meant for the protein.
Right, so again, whether or not your gene is going to be expressed.
And also the binding site, like if you modify the binding site of the protein.
If you modify the binding site of the protein, so do you mean modify the transcription factor so it cannot bind to DNA?
Yes.
Yeah, that's another one. So if you phosphorylate your transcription factor or modify in some way, then you're not going to be able to, you're going to change your binding site. So that's another one. So now we have 3. Yeah.
Like, also we can think about the pathways, like the way upstream, like, let's say, I don't know, LAMP kinase or PLC or
Right.
like G-coupled receptors.
Right. Okay. So up in the signaling pathway, you can regulate whether or not your transcription factors are going to get it in through different mechanisms. So I want specific mechanisms. Yeah.
Whether or not the ligand binds to cell surface.
Whether or not your signaling pathways ever activated. Right. So if your signaling pathways never turn on, then your transcription factor is going to stay in the cytoplasm. Right? Okay. Good. You guys are all on the right track. Yes.
If it's tagged for degradation.
Okay, stability of your transcription factor. If your transcription factor is degraded, it will not be able to enter the nucleus and bind. Yep. Yep. Phosphatase. Okay, so then that's the phosphorylation status, right? So whether you're in some cases phosphorylation might be inhibitory, some cases phosphorylation might be activating. So there, yes, phosphatase activity could have an effect. Yep. Any other questions? Any other thoughts? Yep.
Upregulation of transcription.
Right, upregulation of transcription factor. That goes back to what we were saying before. Whether that protein's ever made, you can regulate that at the level of transcription, at the level of translation, at the level of RNA stability. Yep.
I know that many people struggle with this translational modification, but did anyone mention that, and if it's possible, I don't know, that one of those post-translational modification can help direct the protein to the nucleus?
So post-translational modifications that send proteins into the nucleus. Yep. Sometimes phosphorylation, or even simulation. There are other modifications that will help to direct proteins to the nucleus. Any other thoughts? You guys are all on the right track. It took a minute to get on track, and once you are, we're good. Should we go through what the book says? Okay, protein synthesis. So that's exactly the first few things that people said. You know, you're inactive when you don't exist. I think that's funny. And then you're active when you do exist. So transcription, translation, yeah. Stability. Ligand binding, right? So someone said something about ligand binding. In some cases, your transcription factor is going to be inactive unless the ligand is bound. We learned about that quite a bit in the bacteria story, right? What repressor did we learn about that's only that's regulated by ligand binding? Trp repressor, GLAC repressor, MET repressor. Yep. Ligand binding. Covalent modification, so that's phosphorylation that we talked about. Addition of a second subunit. So I don't know if anyone said that, but protein, I did hear someone say protein-protein interactions, right? I heard someone say that. Sometimes you'll have a second protein that needs to bind that will allow for interaction with DNA.
Is that is a case of yeast?
With yeast and GAL4, Kind of. Are you talking about what I haven't taught yet today? Did you listen to the weekend lecture? Okay. There is actually, in yeast and GAL4, you do have an inhibitor that often binds. Yeah. Well, that was VP16. So that was like a Frankenstein GAL4. But so unmasking. So that's where you have an inhibitor. This is NF-kappa B. You have an inhibitor that normally binds and masks the nuclear localization signal, so that excludes the transcription factor from the nucleus. But when you get rid of that inhibitor, it allows to go into the nucleus and activate gene expression. Again, another one is stimulation of nuclear entry, so that's another inhibitor. So you can have an inhibitor that's in the nucleus that's going to mask a binding site, or you can have an inhibitor that's going to mask an MLS. And then the other one, which we haven't talked about in all of this class, but we do talk about in cell bio with Notch and Delta, where you can have a protein where your transcription factor is actually in the plasma membrane, and upon activation of signaling, it gets cleaved from the transmembrane domain and can travel to the nucleus. So those are all different mechanisms. Yeah. Ligand is just some protein that something that binds. So that's like when we talked about the TRP repressor. Tryptophan is the ligand that binds to the repressor that changes the conformation. And that controls the conformation. Whether or not tryptophan is bound controls whether the repressor can bind to the DNA. Does that make sense? Okay, so this is all different. You guys got like at least like 3 or 4 of them right off the bat, so that's pretty good. Okay, so one gene might be regulated by multiple regulatory proteins binding to multiple DNA elements within a control region that is 100,000 base pairs long. So it's fairly complicated. And so I'm just giving you all these different pieces of information so that one day, when you're a grown-up scientist and you have to figure out how a gene regulatory protein works, you can go back in your mind and be like, well, could be this, could be this, could be this. Then you design experiments and you kind of can study these things. Okay, so I've got, I guess, one example of regulation of initiation. Oh my God, where's our time going? Who's that? No.
Sorry.
It's like being the nurse in the hospital. That's what I realized. The nurse in the hospital is loved by like everyone but the patient. Because the nurse always comes with like the needles. And they're always hurting you. It's so sad. It's well actually, you know, and actually in the pediatrics, the nurse is not allowed to do anything with needles. They actually have like the people in different color scrubs come in for anything needle oriented because they don't want the kids to be scared of the nurses, which is kind of nice, right? Whereas like in the adult world, it's just like, here we come. So, but sometimes I feel like I have to be the evil person. So I make you sit here and listen to me talk for hours.
You can become the good person by giving everyone
I know. If I become the good person by giving everyone an A, then I become the unemployed person.
You want to be here.
I'll be on the good one. I know it's true. It's true. I know you guys are the good ones. Your chair. That whole last row, just like in the last few weeks, they just disappeared. Like there used to be like I even know who sits in each seat. Do you think they're on spring break?
No, winter break.
You're just stuck on winter, huh?
It's cold. It's cold outside. It's really
It got, oh no. Paint top on. Okay. So let's talk about ELK1. So ELK1 is an activator. These are just examples I'm going to give you guys. Don't memorize them. Just think about them. ALK1 activates transcription in response to mitogens. Mitogens, you guys remember mitogens? We talked about them with cell cycle, right? These are growth stimulators. And the way it works is it directly recruits mediator and HAT. Makes sense, right? Right, so in the absence of mitogen, ALK1 is going to be bound to the DNA actually, but it's going to be in a conformation where it's inactive. And then what happens is if a growth factor, mitogen of some sort binds to a receptor and you get signaling, then you'll get phosphorylation of ALK1 in its activation domain by MAP kinase. And that changes its conformation. So notice it goes from this thing to this thing. Who used to play Tetris? Who plays Candy Crush? Yeah. So you like, I feel like sometimes like it's all about like putting pieces together. Anyways, so it changes to this shape, and then that allows for different proteins to interact. So it recruits mediator and HAT, and then that's involved in activation of gene expression. So that's one example of regulation of initiation. Yes.
The mitogen causes the phosphorylation changes the conformation to active.
Yeah, but the mitogen is outside the cell.
So it comes in the cell.
It binds to a receptor and activates signaling pathway. So that's, go back to this picture here. Oh, go back to that picture there. So here your mitogen would be this growth factor binding to a receptor. And then you have this whole signaling pathway. And in the case of ALK1, MAP kinase ends up phosphorylating ALK1, which changes the conformation, allowing for recruitment of HAT and mediator, and that encourages activation of gene expression. So that's one example of regulation of initiation. So here's another example of regulation of initiation. So this is GAL4, so this is what I thought you were talking about. So this, in the absence, so this is GAL4 and the UAS in yeast. This is about regulation of genes required for galactose metabolism. So GAL4 is regulating expression of these genes. So normally, when there's no galactose, you're going to have GAL4 binding to the UAS, but it's going to be bound by an inhibitor called GAL80. And when galactose levels are high, GAL3 is going to bind GAL80, so it takes it away. And so that allows for GAL4 to interact with SAGA and mediator, these co-activators. And mediator, which then recruit general transcription factors. So here you can see you've got recruitment of TATA binding protein, and then polymerase and activation of gene expression. So here it's slightly different, where you have your activator normally bound by an inhibitor, and then when the conditions are appropriate, you're going to have another protein that's going to compete for interaction with GAL80. And GAL3 actually, what it does is it binds to GAL80 and exports it out of the nucleus to the cytoplasm. So now it's really not available to bind and interact with GAL4, and that allows for gene expression to turn on. So this is regulation of the human interferon beta gene. Interferons are involved in the immune response. And here you can see you actually have cooperative binding of many different regulatory proteins or activators. As you can see, some that are probably a little bit familiar, maybe. You've got different NF-kappa B. You've got NF-kappa B, you've got June, you have ATF. You may have heard of some of these before. Either way, they're all different types of transcription factors, and they all bind to the DNA. This is showing how they actually bind to the DNA. Remember, we talked about these different structures, these different DNA binding motifs that are present inactivators and transcription factors. Well, so here's your DNA, and you can see a coiled coil binding the major groove here, and that's C-Jun and ATF. And you can see IRF, the different IRFs binding. And if you look closely, I think you see helix turn helix. I'm not 100% sure. But you notice they're all sitting right there in the major groove. Here's P50, P65. I think I see some beta strands sitting there. And these are binding to the DNA. All of them binding, just this is direct relation to what we talked about with all those different DNA binding motifs. Yeah.
I don't know if this one believe that, but how, by looking at that, how can you tell where the major group and the minor group is?
So how can I tell where the major groove and minor groove is? Okay, so see right here, that's a little v. And see how that's a bigger v? Little v. So it depends, like that's a much larger space. It's not perfect, like I might be wrong. But when I see, so like usually your major groove is going to be like that big, your minor groove is going to be like little, and then the next one's going to be much bigger. And so you'll see this is the way this DNA is, it's not perfectly straight, it's kind of bent a little, so it's not ideal. But if you go back to those earlier slides where we were looking at the DNA, you'll notice it'll be alternating major, minor, and you'll see that there's differences in width.
Actually, in the end, it shows really good. Like the last.
Like here?
That's a minor, you can say.
This is minor, and then, yep, and that's major. That's much larger there. Yep. And the point of this is, well, one, so you can start to connect all those different DNA binding motifs and those structures that bind to DNA and how they bind and how it might look if you see all these transcription factors assembling on the DNA. And then also to drive home the point that gene expression is off unless all of these components are binding. So that's really stringent regulation, right? It's not just like this combined or this combined or this combined. It's actually all of them. This whole committee needs to be present in order to make the appropriate binding site for then recruitment of all the other activating proteins and coactivators and nucleosome remodeling complexes and HATs, whatever else needs to be present. General transcription factors, everything, in order for gene expression to occur. And it kind of makes sense that you need so many different components present because you don't want to like, oops, inflammation. Like, you don't want like, just like, oh, immune response, oops, sorry, there was no virus there. There's not like, there's no signal, let's just, oh, God, a whole lot of inflammatory mediators. So it's important to have tight regulation. Any questions? I always like this figure. I think this figure is out of a different textbook, so I'm sorry if you don't have it. Okay, so gene expression is constantly changing in response to cellular conditions. And so we've had a few different examples of cellular conditions. We talked about galactose, we've talked about mitogens, we've talked about viral infections. These are these different cellular conditions, right? You also can have an effect by hormones, right? Because hormones bind to steroid hormone receptors, and in the case of steroid hormones, they're hydrophobic, so they can pass right through the plasma membrane and go into the nucleus, where they're going to actually bind to their receptor. So there's a nuclear receptor, bind to the hormones, and then they can go into the nucleus and bind to the DNA and interact with coactivators and activate gene expression. So this is kind of different from what you've normally seen in this class, where I keep talking about these receptors that are on the cell surface. But this is how hormones work. Well, steroid hormones, they actually can diffuse through the plasma membrane and bind to these nuclear receptors that then change their conformation, enter the nucleus, and bind to the DNA and activate gene expression. Okay, so here you're seeing that, wait a minute. So it could be two different ways, actually. So either you can have your receptor bound to your DNA, bound by a by a co-repressor, and then when your hormone binds, then you'll lose your co-repressor and recruit a co-activator, or you can have your nuclear receptor in the cytoplasm, and then you'll have your interaction with your hormone, and then it'll enter the nucleus. Either way, either one case it'll be repressed or not interacting with the DNA, and the other case it'll be interacting with the activator. And then also this is the NF-kappa B pathway. So here you're having gene expression induced by binding of signaling molecules, activating signaling pathways. And here you're going to have NOS on your P65, which is your transcription factor, and it's going to be exposed as soon as an inhibitor is degraded. And so then it can go and enter the nucleus. So this is one of the things we talked about earlier, where if you're out of the nucleus, you're not going to activate gene expression, but if your NOS, your nuclear localization signal, is exposed, then you can be brought into the nucleus where you can activate gene expression. Okay, so that's all still initiation. So then there's a few examples. You can regulate elongation, right? And so this takes advantage of early stalling. You can have promoter proximal stalling or abortive initiation, depending on which you're talking about, where you'll basically kind of be set up for gene expression with your transcription factors already bound to your DNA, and you're ready to go, but you're stalled. And so HSP70 is an example of regulation at elongation. So normally if you have heat shock, it's a stress response that's going to be induced, and you're going to induce expression of genes such as HSP70. And that is a chaperone that's going to bind to proteins and help to prevent aggregation of misfolded proteins, which you're going to get a lot of with heat shock. And so if you have heat shock, it activates expression of HSP70 genes. And what it's doing is converting stalled RNA polymerase. So you've already initiated, but now you're stalled. And so it's converting stalled RNA polymerase into an elongating complex. So in the absence of heat shock, your heat shock factor is so you'll normally have your RNA polymerase bound, but it's just hanging out, and it needs your heat shock factor to bind and recruit mediator to activate it so we can have transcription. And so it's kind of poised and ready to go. And so when you have heat shock, you get trimerization of this heat shock factor, which is a transcription factor, and it's going to bind to this heat shock element and recruit mediator, and then you're going to get phosphorylation of your C-terminal domain of your RNA polymerase, and it will be converted into an elongating RNA polymerase. So at normal temperatures, your chromatin is open, and your RNA polymerase can bind, and you'll begin transcription, but it stalls. But once heat shock factor is activated by rising temperature, you get trimerization, so three heat shock factor molecules are going to change conformation and bind to each other, and then they're going to bind to a heat shock element in the DNA, recruit mediator, and recruit the kinases, your general transcription factors, to phosphorylate your C-terminal domain of your RNA polymerase, and then you get elongation of transcription. So this is an example of regulation at the level of elongation. Questions? Okay, so then you can also have regulation of termination. So this is called attenuation, and I feel like someone teaches this. Because when I taught it last year, a lot of people came in knowing this already. So you'll have to let me know if this is familiar or not. And so this is a way to control transcription. It's often used as a way to control transcription of enzymes involved in amino acid biosynthesis. And the effects are mediated by RNA secondary structures, and I'll show you what I mean. So this is in regulation of the tryptophan genes. So these are the genes that are associated with transcription and transcription of genes associated with biosynthesis of tryptophan. And so in low tryptophan, what happens is your ribosome stalls because you've got this leader sequence in your RNA. So your first few codons in the RNA are, remember, this is regulation at termination. So you're thinking, wait, RNA? So your first two amino acids, your codons in your RNA, encode tryptophan. So it's a sensor for the amount of tryptophan in the cell. And so you contain these trip codons. And so when you have low tryptophan, the ribosome stalls. And so this right here is showing the ribosome bound to those tryptophan codons in that leader peptide, that leader sequence. And if there's not enough tryptophan to add, it's going to stall. And so that allows this hairpin to form with the remaining RNA. And then it covers up a terminator, and that allows transcription all the way through the operon. This is in bacteria. All the way through the operon, and you get transcription of these genes that are for tryptophan biosynthesis. And with high tryptophan, there's plenty of tryptophan to be added. So when the ribosome is translating, it just adds tryptophan, no stalling, and so you don't get this stem loop. What you actually get is a different structure, which is an attenuator structure, and that stops transcription, so you don't get transcription. You don't get transcription through the whole operon, and so that results in termination. And so this whole mechanism of regulation is dependent upon the rate of translation by the ribosome. This is bacteria, right? Only in bacteria can you have transcription and translation happening at the same time, right? Do you have that at all in eukaryotic cells? No, because they happen in very different places. Who is raising their hand? Yeah. In this system, this mechanism of attenuation is only happening in prokaryotic cells, and that's because it's dependent upon the ribosome, the speed at which the ribosome is translating those first 3 tryptophan codons. Questions? No? Okay, well, we're going to start with this on Thursday, and then hopefully finish everything.